The first clues were found in Mendel's garden. In a quiet monastery garden, Gregor Mendel tended pea plants and observed how traits passed from one generation to the next. He discovered the principles of dominant and recessive traits, laying a foundation for genetics that would resonate through time. His gentle experiments on plant height, seed color, and flower shape revealed predictable patterns, a 3-to-1 ratio that seemed almost poetic. These early revelations hinted at hidden factors we now know as genes. Hello, dear listener. Tonight we embark on a tranquil journey through the history of DNA, the molecule of life. With soft whispers, gentle pauses, I invite you to breathe deeply and unwind. Together we will explore each milestone in a calm, lullaby-like narration, guiding you through the emergence of DNA from obscure beginnings to its modern-day marvels. Swiss chemist Friedrich Miescher worked with discarded surgical bandages, washing away pus to isolate white blood cells. To his surprise, he found a phosphorus-rich substance he called nuclein. He sensed its importance, yet it took decades for the scientific community to appreciate that nuclein was the very substance carrying hereditary information. In 1900, 3 botanists, Hugo de Vries, Karl Correns, and Erich Schermach, independently stumbled upon Mendel's work and reaffirmed his laws of inheritance. This rediscovery, often overshadowed by controversy over credit, ignited a genetic renaissance. Now, Mendel's pea-plant ratios guided scientists toward the physical basis of heredity. Physician Archibald Garrett studied a rare disorder, alkaptonuria, and recognized its Mendelian recessive inheritance. He proposed that diseases might result from inborn errors of metabolism, a quiet revolution linking genes to human health and genetics to medicine. By the 1940s, molecular geneticists speculated that proteins carried genetic information. Then, Oswald Avery, Colin MacLeod, and Macklin McCarty purified the transforming principle, demonstrating that DNA, not protein, could transform bacteria's traits. Their slow, meticulous work sparked a subtle tremor in biology, revealing that DNA held the secret script of life. Biochemist Erwin Chargaff analyzed DNA from diverse organisms and made two profound observations. First, the quantity of adenine equals thymine and guanine equals cytosine. Second, these ratios vary among species but hold true within each organism's cells. These Chargaff's rules whispered the code of the DNA double helix, foreshadowing its elegant symmetry. At King's College London, Rosalind Franklin and Raymond Gosling captured photograph 51, a paracrystalline X-ray diffraction image of DNA's B-form helix. This enigmatic X pattern, gently glowing on film, portrayed the DNA's two entwined strands, phosphates on the outside, bases tucked within, and 10 nucleotides per turn, a revelation that would guide structural breakthroughs. Inspired by Franklin's images and Chargaff's rules, James Watson and Francis Crick put a three-center, right-handed, double helical model of DNA at Cambridge's Cavendish Laboratory. They whispered their discovery in Nature on April 25, 1953, unveiling the blueprint for heredity. Two complementary anti-parallel strands held together by hydrogen bonds between A-T and G-C base pairs. DNA's double helix unwinds at replication forks guided by a helicase and tosperimerase as single-strand binding proteins stabilize exposed bases. Primase lays short RNA primers, which are extended by DNA polymerase in a 5 to 3 direction on both the leading and lagging strands. Okazaki fragments form on the lagging strand and are ditched together by DNA ligase, yielding two DNA molecules, each with one old and one new strand, a semi-conservative dance of duplication. DNA is the universal code of life, transmitting traits from parent to offspring. Segments called genes dictate protein synthesis through transcription and translation. The core tenets of Crick's central dogma: DNA, RNA, protein. Mutations, random or included, introduce genetic diversity, driving evolution or causing disease. DNA stability and capacity to store vast information revolutionized biotechnology, genetic engineering, and medical diagnostics. DNA can conduct electricity. Under certain conditions, DNA behaves like a molecular wire, conducting electrical signals along its stacked base pairs. You have viral DNA in your genome. Around 8% of human DNA comes from ancient viruses that integrated into our ancestors' genomes over millions of years. DNA has multiple helical forms. Besides classic B DNA, there's also A DNA, shorter and wider than B, and C DNA, a left-handed spiral that appear under different conditions. DNA can form a four-stranded structure. Scientists discovered a rare structure called the I motif, a four-stranded knot of DNA that may play a role in gene regulation. Most of your DNA doesn't code for proteins. Only about 3% of your DNA consists of genes. The rest regulates gene expression or has unknown functions. DNA can survive for thousands of years under the right conditions, like permafrost or amber. DNA can remain intact for tens of thousands of years, key to studying ancient species. Your DNA is constantly mutating. Each of your cells experiences about 1,000 DNA mutations per day, but most are repaired before causing harm. DNA is used in nanotechnology. Scientists use DNA to build nanoscale structures like origami, logic gates, and even tiny robots for medical applications. You share DNA with bananas. Humans share about 41% of their DNA with bananas, highlighting the deep evolutionary connections among life forms. DNA can be used for data storage. Researchers have encoded books, music, and even movies into synthetic DNA. 1 gram can theoretically store 215 petabytes of data. Some genes can overlap. In certain cases, one stretch of DNA can encode multiple proteins, depending on where transcription begins. DNA has grooves. The double helix has major and minor grooves, which are essential for protein binding and gene regulation. Your DNA is longer than you think. If you unraveled all the DNA in your body, it would stretch from Earth to the Sun and back over 600 times. Identical twins are not truly identical. Though they share the same DNA, environmental factors and epigenetic changes make them genetically unique over time. Some DNA is junk. Or is it? Once dismantled,